Immune response to the antigen usually requires that the antigen be recognized by a T cell, right? Because that's what I started talking about. This is an example of where the antigens come from. Different bacteria, viruses, and so on from your environment. Now, if different antigen in the B cell environment, let's say these are all different shapes of antigen. If this is different, the B cell that has this can bind any one of these, right? It's not stuck to be binding only the autoantigen. And what an antigen does, if it comes into a cell, let's say it's a general antigen for a mutated person that can bind different ones, it gets processed, it's a complicated process, and then it gets expressed by the class 2 major and by the antigen presenting cell, and then at least it kind of recognizes the T cell receptor and it does something with the T cell, right? This is what's supposed to happen normally. And the T cell can then turn on genes, and then it can make the B cell do a lot of other things. See the antigen right here? It can bind a different antigens. Now, this gets complicated because all of these antigens that bind, that kind of bring the immunoglobulin molecules together, and then they make more of a response. So like over here, the antigen can bind, and then they kind of get together like a little group of the IgM molecules kind of move together, and then they can make more of a response. This is just background stuff on how this antigen binds to the receptors. See how an antigen, it can bring this together and then can elicit a response. This is your heavy chain and your light chain. This is your constant region that goes underneath the cell. Now, I just put a bunch of pictures here because you can kind of have to understand that once an antigen, depending what antigen it is and what antigen binds determines your mutated sequence or not, it can do a lot of different things inside the cell once it triggers the signaling capacity of that B cell receptor. Okay, and the T cells, if it happens, it does plasma cells and memory cells, and things change. This is a regular immune response. Now, when an antigen B cell receptor binds, again, this is just similar things. It gets internalized and gets broken up, and then it gets presented to the T cells, and you have T cell response going on. Now, and the T cell changes. What happens is if a B cell is not mutated, it binds a different type of antigen than the antigen from a mutated CLL cells. And mutated CLL cells can bind many different kind of antigens. At kind of like, it's better because it doesn't bind autoantigens. Autoantigens are the ones that bind to the germline sequence because that's a different shape that can recognize the autoantigens better than if you mutate. If you mutate it, you have mutations there. You're not going to recognize your self-antigens as well. Right. The normal B cells have evolved to be this way. You don't want to recognize your own autoantigen because then you're going to become change your. You're going to have these autoantigens there all the time that's stimulating, and the cell is going to make a different type of protein inside the nucleus and the cytoplasm than if it was a mutated binding cell that binds different. So depending what antigen you're binding and how tight these bindings, different genes and kinases and things are turned on inside the cell, which will either turn on growth factors or anti-apoptotic or apoptotic genes, depending what this initial stimulus is. This is all different depending if you're mutated or not.
So let me right. Say something and see if I'm right on that. So the reason that being unmutated is bad is because you have your CLL cell, and if it's unmutated, it continues to do the wrong thing over and over again.
Yeah,
but if it's mutated, then it can pull in other things.
Well, I mean, it's still, they still have CLL even if you're mutated. You just have more of an indolent. Normal right. B cells are usually mutated. The normal B cell is supposed to be mutated, a normal good B cell, because it can bind many different things. Right. Little antigen, not autogen. You want it to be mutated. You can bind a virus from your flu or influenza or bacteria, and then you can elicit immune response with the T cells and stuff like that. Because that's a different signaling than an unmutated cells, which still remained in its germline sequence. Because see, what happens is, and I don't have time to go into all the maturation of a B cell. No. There is a B cell here. Where's my mouse? Okay. So you have a B cell, and it finds an antigen. Where is it? So there's now a cell that by chance goes into this part is called the germinal center. The germinal center is part of it's in your body. If the B cell goes through there, it becomes mutated. It finds it. There's different antigens yet, and then it comes out to be a mutated CLL cells, and it doesn't really bind an autoantigen as well. Has an unmutated CLL cell that does not go through the germinal center. So these two cells have a different fate. This one, which is a good CLL B cell, is known to go through the germinal center, get mutated because that's what happens in the germinal center. It sees several different antigens, and then it'll have variation here, which it can bind to antigens in the environment, and the signaling genes turned on are different than these genes that are turned on in here, which doesn't go to the germinal center, and it's more like a different antigen because the shape is different. It's more like an autoantigen, which makes the cell have a poor prognosis and turn on different genes that are involved in anti-apoptotic, not to survive longer. So it really has to do with the shape of the B cell receptor, how it looks, whether it's mutated, and mutation changes the shape. So for example, an unmutated, yes?
Where do the antigens come from?
We don't know what they are. I mean, of course, we wish that's kind of the holy grail. We don't know what there's a lot of labs that are trying to figure out what are the autoantigens in here versus these antigens, and it's really not. It's a very hard field because people have tried to take this immunoglobulin and separate it for the cell when it's bound to the antigen, trying to separate the antigen from the immunoglobulin molecule, and it's very hard research because a lot of things change if you do separate them, and it gets degraded. So there's several papers out, but we still don't have an answer to what the actual some of these autoantigens that they think as like bacteria, like in your gut, in your body, that your body kind of produces, and by chance this cells with its germline sequence bind this antigen X much better than someone else, and that antigen selects for these B cells. It turns on different genes in all their signaling pathways, and those genes make that cell resistant to apoptosis, and it gives the patient a worse prognosis. So it's random whether you have, what VH sequence you have here, and whether the antigen, if the cell is exposed to an antigen. I mean, because, you could have these cells and you don't have exposed, and that cell doesn't get stimulated and maintained in the environment. So basically, it really has to do a lot with the shape of the immunoglobulin on the cell surface, which determines what kind of signaling pathways are turned on or not, and the shape again has to do with the mutation. So in this case, you want more mutation. And also, let me just go, like for example here, let me go here, it gets kind of complicated. This is an unmutated CLL cells. And remember, the immunoglobulin molecule is still in its germline sequence, like the original sequence. It hasn't been mutated. So it sees some antigens. So these cells, because of the shape, get activated. There's different signaling pathways that are turned on. The antigens are still in the environment for some reason. We don't know what they are. So these now becomes a clone. It becomes really a tough CLL cells. It has different genes turned on and off. And then if it stays in the body, sometimes it becomes a predominant clone, and sometimes we get subclones, but it's not very common. Instead, in the mutated CLL cells, it sees different antigens. They're not the autoantigen, the same ones that unmutated sees. And some of these cells, because those little antigens are not always around, they become anergic or inactivated, and some of them kind of die because the antigens are kind of turning off genes. And then some of these, the antigen is still on, and then it becomes a different clone, but this clone D is different than this clone A, has different genes turned on and off inside all their signaling pathway, and those genes happen to make that cell more benign versus the genes that have been turned on this cell. The other thing is that we're finding nowadays, you know how all these other, like SF3B1 and Notch1 mutation, I don't know if they know about, all these other, there's a lot of cancer genes that are being sequenced nowadays. And they say that some CLL patients have higher increase of these other mutations. Well, these kind of cells here, since they're like hardy and they're like more tough, they also can accumulate these other cancer mutations. Even though you said, well, wait a minute, he's unmutated, but now he's getting mutations in other genes, not the immunoglobulin gene. It can get mutations in other genes that this cell does not get. So those accumulate, or this cell now that it's a different type of cell than this can acquire 17p more, or trisomy 12. This cell, since it has seen different antigens and it's kind of more because it's mutated and it doesn't see the outer, it's not such a bad cell now because it's just like kind of hovering there. Does not get additional bad mutations like 17p or Notch1 that this cell does. So, all of this, of course, doesn't happen overnight. It takes years or an environment and many things for it to happen. And so by the time we see the blood, it's basically at this point, and we're doing VH sequencing at this point in the whole life cycle of the B cell.
I have a question. Is the mutation constant when you first get CLL,
yes,
or does it change?
It's like set in stone. I can take a sample before and after treatment, redo the VH, and it's the same. This is set up here, the very beginning, in the germinal center, remember or not. This is the very early, doing the early B cell development. And it's just set, it does not change. Like if you get treated, that of course your B cells will go down, which is great. And if some of them happen to not get killed by the chemotherapy, and it's called minimal residual disease, you probably know, MRD.
Yeah.
So we would like a patient to be MRD negative. That means minimal residual disease negative. You don't have any minimal residual disease. If a person has been treated, and let's say he has some minimal residual disease, if we're lucky, we get the cells and we make the DNA, those few, let's say you have, 50 CLL cells left over in MRD, if I do VH on those, they're going to have the same exact VH that you had 5 years ago when you had 1,000 CLL cells. That VH, which is kind of not nice, but it's kind of good for us because it's almost a signature of that patient and that cell. And we can say, well, if we still see VH there, it's not a good sign because there's still some CLL cells. And that VH, let's say you're VH169, that will still be VH169. It's not going to be VH2 or VH3. It doesn't change. And so it's nice as a biologist because we can kind of follow where that B cell goes by their VH signature, but it's not good for the patient because you want it all to go away after chemotherapy or
I have
things
one
like
more
that.
question.
Yeah.
Can you have too much mutation?
Too much? I don't, I don't think we don't get most of our mutated patients are 10 or 20% different from the germline. It doesn't go to all different. No, I don't think so, no. But I don't know, most of our patients are, I'd say they're 85% homologous to the germline gene. That means it's 15% mutated or 20% mutated. So there's probably a threshold. I don't know, how these mutation mechanism. It's kind of complicated. There is an enzyme that's called AID, and that when the DNA gets opened up in the germinal center, it replicates and it makes mistake, and there's enzymes that either fix them or don't fix them. So it depends if the enzyme, you have a mutation in that enzyme and it doesn't fix your mutations. But usually there's not a lot of mutations. But it does make your cells better because it binds different antigens that are not autoantigens in the environment. And so I try to make a little summary here. And please tell me if it gets too complicated. And so let's say, okay, I'm asking the same question again, mutated VH genes. So, but don't forget that having a mutation is not necessarily pathogenic. Normal B cells have mutations in their VH genes because you want your B cell to recognize all the little antigens and pathogens in the environment and elicit immune response with the T cells and everything else. And also in the the VH gene, if it has a mutation, we know is that needs to occur in the B cell to do its job to bind to foreign antigen, which I said. So somatic mutations, if they have occurred, that means they're from the germline VH sequence. This is a normal process during B cell development, and this is an indication to us that that B cell has passed through its life cycle through the germinal center, because that's where their mutations occur. Then normally you need this variability in the VH repertoire to bind to several antigens. Now VH mutation can decrease binding to the autoantigen, which you don't want, right? Because it doesn't fit, because now they're different. So that's good. And each mutation can enable the cell to bind these other antigens in a different way. And of course, depending what antigen you're binding, the B cell receptor signaling cascade inside the cells, like I showed you in the little diagram, is going to turn on different genes in this one than the unmutated people. And then this can result, because of what the proteins are turned on and off, in a more indolent disease. So you would have a low probability, these B cells, to accumulate either genomic aberrations, like I said, like Notch1 or these other new ones that are coming out, or cytogenetics that could be related to cancer, that are not going to make your disease get worse. There's low probability of that happening. Therefore, these patients have shown in the clinic with their, like I showed you, the survival curve, they have a more indolent course. Now, in the opposite situation, where you're unmutated, these sequences are still in the germline conserved, not mutated. That tells us that these B cells has gone in a different kind of course of its differentiation, has not passed through the germinal center because it's still not mutated. And therefore, it has the potential to bind a different type of antigen than the other one that was mutated. And therefore, if it binds a different antigen, then there's a different signaling, different cascade, different genes turned on. And unfortunately, the ones that are turned on are anti-apoptotic resistant, and they change things in their clinical course. And again, unlike the other cells, since these cells have a growth advantage, because these proteins, different proteins that are turned on, these cells have, unfortunately, can accumulate other genomic aberrations related to cancer. That's why many times people that have unmutated VH genes are also high probability of having 17p or trisomy 12. If we took 1,000 patients and look at where the 17p deleted people, most of them have unmutated VH. Again, it goes towards up 70 positive and so on. And it's all involved in the prognosis of the patient. So this is, hopefully you kind of understand a little bit better why this happens clinically. And I have to really thank everybody in the CRC, the CLL Research. This is Dr. Kanti, right? Dr. Pluckett, and Dr. Kim. We're all a lot younger. This is taken. I was going to say, oh God, I should just only use this picture. This is Bill Weird at MD Anderson, and Gribben in England, and Dr. Carlo Croce, that he does a lot of microRNAs. I work very closely with him, and Neil Kay at Mayo Clinic, and then he's Dr. Griever at Ohio. So you've probably heard of a lot of these very famous doctors throughout the CLL literature. And of course, the lighting is not very good. This is actual Dr. Kipp's lab that I have to thank everybody. And actually, Tuan and Claire are here in the audience. They're my two helpers. Tuan is doing a lot of the VH sequencing, and Claire is an intern student, pre-med, hopefully, for working in the lab, and all of these other people are upstairs working hard. I'm sure. So, and of course, I really, really have to thank all of you patients, because if it wasn't for you, we really would not even have a lab, and everybody in the CLL world really has to thank all of you and your dedication, and we really appreciate your contributions and trying to help us out in some of this, understand the biology of these B cells. So I really appreciate all of you.
Thank you.
Oh, and I have another funny picture that I always try to make an analogy. It's like, why are you freezing so many cells, downstairs? I've got like 500,000 vials and stuff. But, and somebody in another meeting gave me this, it's kind of like a wine cellar, A wine gets a lot more expensive as it's older because the quality, and I'm not even a wine drinker actually. But so we figure that CLL's vials in our tissue core repository are very, very valuable because as a patient, you know, we have the cells and we find new ways of looking at the DNA or making discoveries. We can go back to those vials and look at the genetics or serial samples of a patient. And I've been doing this 2000, so it's like some of these vials have been sitting in our repository like for 13 years, and so we can go back and study longitudinal or redo the same vial again with a different technique and maybe understand the biology of this disease even better as technology advances and it gets better. So we're trying. So I thought that was kind of neat. Sorry, that's all. I really don't have to. Thank you very much. Yes.
Did what happened at the University of Pennsylvania a couple years ago with the 3 patients